pH method. In aquatic ecosystem, the pH of the water is a function of the dissolved carbon dioxide content, which in turn is decreased by photosynthesis and increased by respiration. However, to use pH as an index to productivity, the investigator must first prepare a calibration curve for the water in the particular system to be studied. Because pH and carbon dioxide content are not linearly related, and the degree of pH change per unit of carbon dioxide change depend on the buffering capacity of the water. Thus, 1 unit of carbon dioxide removed by photosynthesis will bring about greater pH increase in the soft water from mountain stream and then well buffered sea water. The relationship break down in those areas, both freshwater and marine, in which photosynthetic activity is accompanied by precipitation of calcium carbonate, and the method obviously cannot be used in such condition. The pH method has been especially useful in study of laboratory micro ecosystems, such as scenes with pH electrode and recorder. One can obtain a continuous record of net daytime photosynthesis and nighttime respiration, from which gross production can be estimated without removing anything or otherwise distributing the community. Disappearance of raw material. Productivity can be measured not only by the rate of formation of material, food, protoplasm, mineral, and by measuring gaseous exchange, but also by the rate of disappearance of raw material, mineral. The method is used in situations like ocean, where certain constituent like nitrogen and phosphorus are not in a steady supply throughout the year but accumulate in water once in a year or at interval. The rate at which the concentration of this raw material decrease become a measure of productivity during that period. Disappearance of raw material. Productivity can be measured not only by the rate of formation of material and by measuring gaseous exchange, but also by the rate of disappearance of raw material, mineral. This method has been used to measure the production of certain ocean where phosphorus and nitrogen accumulate during the winter and then used for the growth of phytoplankton during spring. The disappearance of the amount of raw material which get incorporated in food production measure the net production of the whole community. This method, however, cannot be used in situation which has a steady state of equilibrium, wherein the amount used might be balanced by the amount being released or entering the system, and there would be no way of determining the actual rate of use by organism. Chlorophyll method. This method is based upon estimation of productivity from chlorophyll concentration and light intensity through the water column. The technique evolved from the discovery by plant physiologists that a close relationship exists between chlorophyll connection and photosynthesis at any given light intensity. With proper calibration, the area-based chlorophyll content of the whole community can provide an index to its productivity. In a given light adaptation system, the chlorophyll in the autotrophic zone self-adjusts to nutrient and other limiting factors. Consequently, if the assimilation ratio and available light are known, gross production can be estimated by the relatively simple procedure procedure of extracting pigment and then measuring the chlorophyll concentration with a spectrometer. The method was first used to estimate primary productivity in large water bodies such as sea, but later applied to terrestrial ecosystem as well. This method involved the determination of chlorophyll content of phytoplankton in a given volume of water. Because all plants need chlorophyll to carry on photosynthesis, the amount of chlorophyll in a given amount of water is a direct measure of the total biomass or standing crop of phytoplankton it contains. The chlorophyll is extracted chemically and amount of chlorophyll is measured. The deeper the color, the greater is the concentration of the chlorophyll and therefore phytoplanktonic biomass. Productivity determination with radioactive material. One of the most sensitive and widely used method for measuring aquatic plant production is done in bottom with radioactive carbon 14C added to carbonate. The theory behind using 14C to measure productivity involved using a label tracer quantify assimilated carbon. The 14C method estimated the uptake and assimilation of dissolved inorganic carbon by phytoplanktonic algae in the water column. The method is based on the assumption and biological uptake of 14C level DIC is proportional to the biological uptake of the more commonly found 12C DIC. The method consists of collecting water sample before drawn in a series of bottles, generally two clear and one amber or dark color bottle, through which light is not penetrate. The water sample are in inoculated with 14C capped and placed in the environment they were collected from. Following the incubation, the sample are collected under light condition and filtered through 0.2 filter. The filter is placed into liquid sanitation vial, acidified parts excess 14C and keep cold until it can be analyzed in scintillation counter. The radioactive count are used to calculate the amount of carbon dioxide fixed in photosynthesis using the photosynthetic equation. The technique has its deficiencies. The estimate is based on assumption that radioactive carbon is proportional to the stable carbon. However, in practice, this may not proportional. Therefore, a correction factor of 14C absorption in dark bottle is required. It does not discriminate between respiration of phytoplankton and bacteria. The uptake and release of 14C by bacteria and zooplankton and the abilities of different species of phytoplankton to use available light. Ecological efficiency, food chain efficiency, trophic efficiency. It refers to percentage of energy transferred from one food chain level to trophic level to another food chain level or trophic level. The calculation of ecological efficiency typically focuses only on the consumer that feed on living tissue, ignoring derivatives, detritivores and decomposers. It can be shown mathematically. A2 divided by A1 gunon 100, where 1 A1 is lower trophic level and A2 is higher trophic level. It follows the 10% rule, which states that roughly 10% of the energy at one level will be available to used by the next level. Ecological efficiency is always less than 100%. There are three reasons for this. Consumer at a particular trophic level may not eat the food that potentially available to them. Two of the food energy that is eaten not all assimilated into body tissue but instead is lost in digestive waste. Ecological efficiencies typically range from about 5% to 20% biomass. It refers to the total dried biomass of the living organism in a particular area at a particular time. Gram per meter square per year. Standing crop or standing biomass. It refers to the total dried biomass of the living organism, such as uncut farm crop, the fish in pond, or organism in an ecosystem in a particular area at given time. The standing crop may vary at different time of the year. For example, in a population of deciduous tree between summer and winter.